new activity for me and one that I really enjoy. And I'm standing in for Russ Bergman this morning, who got called away at the last minute.
He's right here.
Oh, there you are.
He's literally
And
right here.
I gave it to you because
I didn't expect to see you, and I had a prepared speech.
So Russ, please step out for a
I
moment.
gave it to you because I felt that you were closer to the subject
I
matter.
thought, well, I got it out of office.
Yeah, you did.
Anyhow, excuse me, Russ.
But I think
I'm
Russ
flying
is
here.
a out of office.
Anybody who knows me knows that this is way earlier than I normally get up. Way earlier. I'll have
No,
testimony
because
on
if I do it
that.
the other way, back.
But today's topic
don't know.
is very timely and one that should be of interest to Jews, since we're going to see a lot more.
Hold it.
Just a little bit introduction before I introduce our speaker. In 1953, Watson and Crick, you've heard those names perhaps, described the structure of DNA, the genetic material. And at that time, I was a kid in elementary school, actually. And
That way.
even the scientists didn't dream that one day we would know the complete sequence of the human genome, which consists of 3 million individual base pairs, 3 billion individual base pairs. I have my editor here, if I misspoke. That's at least one of the reasons for having children. And many of you, especially the younger ones, probably learned in junior high school that DNA is made up of pairs of A's and T's, adenines and thymines, guanines and cytosines. And in about 1980, the idea of gene therapy in humans, it started to get a lot of press. It was the cover of Time magazine, the cover of Life magazine, remember those? Mm-hmm. And people started to think about how that could happen, and there were terrible failures along the way. People actually died. There was a famous case at the University of Pennsylvania. So meanwhile, genetic manipulation was revolutionizing agriculture. It was revolutionizing animal husbandry. But humans were a bit behind. In recent years, there have been a number of technological developments, at least one of which we'll hear about in a moment, some of which are actually engineering developments, the development of machines that can sequence DNA at a very high throughput. Nowadays, you can get your whole sequence of your whole genome done for less than $1,000 in a few hours. But the work of genetic manipulation, I'll just say 2 things. It's not trivial, and it's very expensive, whatever you do. So many of you had to read Aldous Huxley's book, Brave New World. And my son probably knows because he's heard me talk about this, but how many of you know where that Brave New World comes from? That comes from Shakespeare's Tempest. And there's the young woman, Miranda, who's on an island with her father. She's never seen a young man, and this young man comes ashore on this boat, and she is just blown away. And she says, "Oh, brave new world that has such people in it." And her father turns to her and says, "'Tis new to thee." And that was exactly right. When you see something like that, it's new and fascinating. And so it is with genetic manipulation. It's new to most people. Its implications aren't widely understood. And certainly, even though I work in genetics all the time, I should have said that I'm a distinguished professor of genetics at Rutgers. Even though I work all the time, I can't guess very well what's going to happen next. It's a good thing I didn't make any bets on the subject. I'd be a very poor man. Anyway, I am so pleased today that we have Dr. John Loike with us, and he serves as Professor of Biology and Bioethics at Torrey University, and is co-director of the Master of Science degree and graduate certificate program in Medical Ethics and Humanities at New York Medical College. New York Medical College, some of you don't know, is located in Valhalla, but Dr. Loike looks nothing like a Viking.
I was born this week.
There you go. And that goes to show you can't determine that by looks, right?
By genetics.
And genetics, yeah. Dr. Loike holds a PhD from Albert Einstein College of Medicine of Yeshiva University, and he did his postdoctoral research at Rockefeller University and Columbia College of Physicians and Surgeons. In addition to being a well-known ethicist, his biomedical research, he's a scientist, focuses on how the human immune system combats infections, neurodegenerative diseases, and cancer. He's internationally known for his work. He's spoken all over the world, including several places in Asia. And he writes on emerging ethical/scientific topics and cross-cultural perspectives. And it's my pleasure to introduce Dr. John Loike, who's going to speak today on altering the human genome, a Jewish perspective. Thank you very much.
Thank you. Jed, Russ, and Dr. Schwartz, is it?
David,
Thank
yeah,
you
yeah.
for this invitation. It's really, for me, this is a real treat, because I usually speak either to undergraduate students, graduate students, sometimes, faculty members, but to speak to it, but most of the time I speak in public forums, but to speak to an educated audience on science is a real treat for me. So I really welcome you to interrupt me, ask me any questions, and if we have time at the end, I'll take any questions on science and Jewish perspective of ethics. So really, because I like to be challenged. I won't have all the answers. Let me tell you a little bit about myself to give you some disclosure. I'm not a rabbi. I, of course, endowed with the Talmud very heavily, but that's a good thing that I'm not a rabbi. But I was trained as a scientist, and it's interesting, I never thought I'd be in this position 20 years ago, because I was focused only doing biomedical research. I was at Rockefeller, at Columbia, I was heavily immersed in the lab, and then in the late 90s, a paper came out on cloning of Dolly, as you all know. I was mesmerized by that paper. I just couldn't stop reading it over and over again, and I realized they forgot to include a very important piece of information. Where did the mitochondria come from? So I wrote to Ian Wilmot and to Campbell and in Scotland, Edinburgh, and I said, send me samples of Dolly, and we'll do it. And we actually happened to do it and published it. When I got the samples, the New York Times came out with an article saying, this is all fraud. Because everybody in high school learned you can't take an adult cell and make it into an embryo and convert it to an embryonic stem cell. And of course, as a scientist, I know the first law of fraud. Never send your samples out. By the fact that they sent me the samples, I knew that they were really correct. And I began reading the New York Times science section a little bit more carefully, and I realized, this may surprise you, reporters don't understand a word of science. So I felt it was my moral obligation to write about science. I began to write about not only what really the science is saying, but some of the ethics about it. And then I started writing with a very good friend of mine, a rabbi, he's a doctor, physician, Aaron Steinberg, who won the Israel Peace Prize. He wrote the Encyclopedia of Medicine from a Jewish perspective. Very internationally well-known guy. And that started a whole new phase of my life where Columbia says, why don't you teach this? So I began teaching a course, which I'm still teaching with all material, that's another story. I began teaching a course called Crossroads in Bioethics, which I go through really science-based ethics. And I'll tell you why. Thank God we have a lot of physicians who who are scholars in rabbinics, conservative Orthodox, Reform. We have a lot of rabbis who are very knowledgeable in medicine. Unfortunately, there are very few rabbinical scholars who are really immersed in basic biomedical science, real basic science. And in those early years, I had a professor at Yeshiva, Rabbi Moses Tedler, who had a PhD in micro. So we decided to write together. And for the last 25 years, we've been writing about 25 papers on the halachic or the Jewish perspective of all these biotechnologies. And what's interesting about Rabbi Tedler, I think, so that's how I got into bioethics. And when I was at Columbia during the last few years, I was director of special programs at the Center of Bioethics under Fishbach. It was wonderful there, but then Columbia, as people Rutgers will know, when you're in the medical school, your goal is to do research and to bring in money. Teaching in India and Thailand and all these places, even at the college, I was teaching at Columbia College, did not bring the medical money, so they said, either you gotta bring in more money or you gotta, do something else. So then I had an offer from Turo, and I left, and I had to give up my lab at Columbia, which was a little bit depressing, but it was, but being at Turo was really very, very exciting because they are very innovative, and they let me design the courses that I want to, the way I want to. I don't have to follow a classical syllabus. I teach genetics, immunology, cell biology, Jewish bioethics, CRISPR, I teach whatever I want, and I can teach it the way I want it, and the students are really excited about that, and I run very interesting programs. One of the programs which is really well known in the New York area is called BioCep, Biomedical Cross-Cultural Education Program. I take students, I've been doing this for a decade, to Bangkok, Thailand, and I show them real-life bioethics. We go to hospital for health and medicine, it's Yankee, and they start out as the center for gender transforming surgery. They can convert any woman into a handsome man, any man into a beautiful woman. It takes a year, and it's unbelievable how they start with 26 beds, now they have over 800 beds. They do anything cosmetic, so whatever you want, they'll do it at a fraction of the cost. They are one of the premier hospitals for medical tourism. They pick the nurses from a beauty contest. How does that sound? And they're all female, and they go around the hospital in miniskirts and rollerblades. I mean, these are the type of things we show them. We take them to an alternative medicine hospital, Jubilee, and they see, alternative medicine. I have to confess to you, as a scientist, I do not understand homeopathy. I don't understand, a little bit I understand about acupuncture, but reflexology, I don't understand the science behind it. And yet I'm forced to engage in this because students say, why don't you do it, And it works. Don't ask me how, but it really sometimes works. So that's very interesting. So we run this program. We're running another one in the Philippines. In Thailand, of course, it's a Buddhist culture. So we have trained a Buddhist, a monk who's unbelievable, brilliant. He's a genius. He's one of the few monks that were allowed to go to university. He was one of the head of the university in Mexico, and I trained him in bioethics, so he gave lectures, and now we're starting a program like this in the Philippines, which is a Catholic, or it was same sort of culture. So this is where I'm up to, and when I left Columbia, I said, I'm leaving. They said, fine, I said, we understand it. But Columbia College says you can't leave because the course that you teach is too popular. So I'm continuing over the last 6 years to continue to teach the course. It's a wonderful course because the students are upperclassmen. These are Columbia students. They're brilliant, and it really is a very challenging thing. So that's where I come from. So I teach at Columbia, I teach at Turo, the graduate school, the undergraduate, and also I'm the first bioethicist who's allowed to teach in China. I was invited several years ago to teach undergraduates in China in the summer, and we have 30 professors from Princeton, Yale, Columbia, and I didn't know this. First time I was over in China, but they rank you. And after the end of the 2 weeks, I was ranked #1 in several years. So I continued to teach Chinese students, It's really very interesting. And at first I could only teach science, but now they allow me to teach bioethics, which means I don't say anything about bioethics. I can ask the students what they think, which is a very different type of situation. Anyways, so that's who I am. I have a book I've written called Science-Based Bioethics. It's a very unusual book because although Cambridge Press and Columbia wanted to publish it, I said, sure, but you gotta revise it every other year. Because the science is so new, and they said, we can't do that, we can only revise it every 5 years. I said, forget it. So I self-publish it, but it's written basically for my students. They all come in, and from Columbia and Turner, they all write. So it's it's in a sense a student-oriented textbook, and they love it, and I think it's great. Anyways, the other thing I want to tell you is that I have to tell you a little bit about Rabbi Kemmer, because he was my mentor in ethics, especially Jewish ethics, and a remarkable, remarkable scholar. As I told you, he has a PhD from Columbia in micro, and he taught me several basic principles in terms of bioethics. Number one, if you don't understand the science, you can't understand the legal, social, or Jewish perspectives. You have to. So every time I tell him about a new technology, he said, I want to see the primary papers. He used to read the primary papers and discuss that. And this is something which, unfortunately, even I was just at a conference in Phoenix on Jewish bioethics and technology, and there are some of the most famous bioethicists. They're great in philosophy, psychology, etc., but they don't know the details of the science. And because of that, there are differences. So that's one thing he taught me. And the other thing is that he was a student of Rabbi Feinstein, who was one of the great Orthodox decisors of Jewish law, and he taught me that when you actually deal with actual cases, you've got to take into consideration the psychological status of who's asking you that. And because of that, it would seem that he should be very strict, but in fact, he was extremely liberal in issuing his rabbinical decrees on a one-to-one level. And that was always amazing. And I'll tell you, his insights into human psychology was very striking. I'll tell you one story, because we have time. Again, you can help. So we were writing a paper on gestational surrogacy. Here's the Jewish issue. You have a couple, and the woman's born without a uterus, happens 1 in 100,000, or she has a defective uterus, she cannot carry a child. So they take his sperm, her egg, they do in vitro fertilization, get a blastocyst, and they're going to implant it into a gestational surrogate. Supposing the gestational surrogate is not Jewish, what do they do? What is the status of the child? Very, very interesting case. As a matter of fact, it's one of the first cases in which there is no precedent in Jewish law, absolutely no precedent about that. So in discussing this, there is a process. If there's no precedent in halacha, in Jewish law, you have to be very strict. So in this case, most of the great rabbis, including Rabbi Kemmer, will say, you must convert the child. Because we don't know, is Jewishness determined by genetics? Is it determined by birth mother? And it's a whole interesting area. And then one of the things he said to me, Rabbi Kemmer, he says, but you have to understand that to do this, you have to be very clear to the couple engaging in this about all the dangers and the possibilities. So what do you mean? He says, well, it may be that the husband is going to fall in love with a surrogate because she's carrying his child. I said, Rabbi, come on, how often is this going to happen? He says, do a literature search, figure it out. So I did, I found nothing on this, but it turns out there was a professor at Columbia, Nancy Reeves, who had done a major study on gestational surrogacy, on the psychological, social aspects. So I sat down with her at an appointment, and I said, just ask her, tell me about all of the stuff that she found. And she says, one of the most interesting things she found is that how often the biological father would have an affair or divorce the mother and marry the surrogate. So here Rabbi Kemmer understood this, which I didn't understand. That's who he was. Remarkable man. And I had him as a biology professor. I have to confess to you, I was a terrible student in his class because he was too bright. I couldn't figure out anything, but that didn't stop us. But the first day, he would put us all in front of the microscope, and he had a slide of a human cell. And we'd look and say, what do you see? And, you know, I see the nucleus, that's the membrane, I see some, looks like mitochondria, I see, you know, all this stuff. And he says, you're all blind. What you should see is the hand of God. This is, if you look at science and you don't see the hand of God, you're not looking objectively. That's what our hand was. All right. With that introduction, we're going to go on to CRISPR. I'm sure I'm going to be a little bit simplified, so excuse me if I go too simplistic. But I think what's really important for this talk is in the last few months, the FDA has approved the use of CRISPR for sickle cell anemia. This is remarkable, and I'm sure many of you know this, but just let me bring everybody up to the par. In sickle cell anemia, you have usually of many, but typically one nucleotide is mutated, and the hemoglobin still functions very nicely. But what happens is that the hemoglobin becomes sticky and forms a fibril structure and becomes the red cell, instead of having a disc, becomes a sickle cell, and that causes aggravation because it gets stuck in all the branches, and you get anemia because you can't keep up with the destruction of the red cells. And these patients suffer from anemia, as you all can imagine. So somebody asked a very almost Talmudic question many years ago. Says, wait a second, this is a genetic disease, hemoglobin, right? Why is it that the embryo survives? There's no sickle cell in the embryo. And they did research, they realized that the embryo does not make what we call adult hemoglobin. They make fetal hemoglobin. Why? Because they has to have a higher affinity for the oxygen to carry it from the mother across the placenta. And after birth, within the first 6 months, the newborn child turns on a gene that turns off the production of fetal hemoglobin to make adult hemoglobin. And sickle cell anemia is basically an adult hemoglobin defect. So they said, let's use CRISPR to prevent that gene from being turned on. And that's what they did. And you could read, it's amazing to read these people who, within weeks, who were suffering from anemia, now their eyes, instead of being yellow, because of all, the destruction, they have white eyes, they don't have to have transfusions. It's an amazing breakthrough. It's still new, I can't tell you what the side effects are, but it's an amazing thing, and it says something profound about CRISPR that the FDA is willing to approve it so quickly. Usually, any of you in science know it takes decades. Gene therapy, we're still just beginning now to get some therapy, gene therapy. Stem cells, we're just in the beginning of stem cell therapy, which is unbelievable. I think it's one of the most exciting things. But I think that, in my mind, CRISPR technology is one of the most brilliant scientific innovations over the last 25 years. And, I should tell you, it's very, as Jay pointed out, it's very expensive, and we'll discuss that in a few minutes. So let's go through. I don't have to tell you, I don't think I have to remind you, DNA makes RNA, makes protein, and all that sort of stuff. But I do want to emphasize a couple of things. The first is that the cell, most cells, especially human cells, have at least 4 primary mechanisms to repair breaks and to repair mutations in the DNA. This is amazing how they do it. And when you teach, you always have to ask, how is it? How do they recognize a defect, a mispairing of bases on the DNA? And it's brilliant. It's like, I guess the best analogy, you have a road. If you have a bump in the road, you know something's wrong. If you have a depression in the road, something's wrong. And you have genes that survey the DNA and look for these bumps. And in addition, as many of you know, DNA carries electrons. And if there's a break in the conductivity of the DNA, these enzymes come into play. And they're amazing in their capacity to repair breaks. And that's very important in terms of CRISPR technology. Now, as I'm sure many of you know, and I'm sorry to repeat this, CRISPR technology, I'll tell you why it's going to be important later. CRISPR technology was discovered basically in the bacteria. If a bacteria is infected with a virus, a bacteriophage, and it survives, it cuts up some of the sequences of the phage, of the virus, and incorporates it into the genome of the bacteria. And if subsequently they are reinfected, it has this RNA guide that recognizes that bacterial sequence. It calls them CRISPR-associated proteins, Cas9, and it forms a complex with the invading virus, and like molecular scissors, just breaks up the virus and destroys the virus. And the idea of applying this technology to editing the holy grail, our DNA is remarkable. So you have various components. You have, of course, the DNA, the Cas9, which is the molecular scissors, the guide RNA, which will target, and there's really two targeting things. There's a guide RNA that's called a PAM. I guess a simplest way to say is the PAM tells you what street the Cas9 has to go on, and the guide RNA tells you the number of the house. By the way, house here are beautiful. So wonderful. I'm sure to say that. So then it causes a molecular break. Now, the cell has to repair the break. Now, if it's a double-stranded break, which often it is, that's a really challenging thing because there's no template. So if you don't have a template, it's going to randomly insert whatever base pairs it wants. That's not good. So what we do now is we give a complementary a DNA, a cDNA, to serve as a template to tell the cell how to repair it and to correct the actual break the way we want it. So you can correct, it's like an editing, you can correct the whole genome. This is what CRISPR is about. Is that clear, everybody? All right. All right. So, what are some of the medical applications of CRISPR? Okay, there are about 1,000 genetic diseases. We can theoretically cure all of them. Many of them are by single base permutations. What's amazing about CRISPR is it's not a one-time, it's a one-process thing. We've done CRISPR for 60 or more genes within a genome at the same time. And we have good targeting, we're in great shape. We'll be able to affect diseases like ALS, diabetes, HIV. I mean, there's a whole host of things that we can do. Xenotransplantation, I'll talk to you later about that. I was invited to, there's an international conference on xenotransplantation in Istanbul. I was there, I was in, I guess it was San Diego 2 years ago talking. They wanted me to come to speak about the ethics of xenotransplantation, taking a pig heart, putting it into a human, what is a Jewish perspective of that. Very interesting. And all these things can be, and to do that, we're creating these genetically modified pigs by CRISPR to make them more human-like. It's amazing. And of course, longevity, right? We can use CRISPR to improve our lifespan. I don't know how healthy it's going to be, but 120 years is not the limit, all right? So there are a lot of therapies that we can be doing. Now, discuss some of them. For example, let's talk about HIV. The simplest application is you take a person who's infected with HIV, you give it a vector with Cas9 and the guide RNA that's going to only target the RNA of the HIV, and now it's going to invade all the cells. This it's a nanoparticle, it invades most of the cells, and if it sees that genome from the HIV, the molecular scissors will be activated, it's going to cut the virus, and if it cuts the virus, you're in good shape. You're not going to be able to infection. That is one example. Another example, phage therapy is, I think, a brilliant idea. They're doing this in the Netherlands. As I told you, bacteriophages are highly specific. They only invade a specific type of bacteria, and they can cause destruction of the bacteria. So what they did is, they said, wait a second, let's give that phage a new weapon. Let's give it CRISPR. So now you take a phage, it has CRISPR, and it recognizes the sequence of a unique bacteria, C. diff, toxic bacteria. So you take patients, they've done this, who have bacteria that are resistant to the antibiotics, you give them this phage therapy, and within 2 weeks, you've really ameliorated most of the infections. It's a remarkable, remarkable technology. having the phage, which only infects the bacteria, doesn't affect any other cell, doesn't really harm, will infect that. So that's another example. CAR-T, of course, as you all know, chimeric antigen receptor therapy. It's a brilliant, I mean, it may have some problems, but it looks like it's, at least for certain lymphomas and leukemias, it's 80% effective. And the idea here again is to use CRISPR to really target your T cells, to make sure that your T cells are going to target the right genetic mutation in the cancer cell to destroy the cancer. It's remarkable. We're going to see more of that. And I told you the transplantation, creating these pigs that are more akin to human tissue. It's interesting, when I spoke about this in San Diego, they said, what is the Muslim? Why do the Muslims object to this? And because unlike the Jewish perspective, it's okay, we can take a transplant from the pig, there's nothing, it's not we're eating the pig, there's nothing wrong with that. But the Muslims have a problem with that. And they asked a very good question. Why did you have to do it on pigs? Why couldn't you do it on sheep, right? We could do it, avoid all the issues. But again, you know, science, as all of you know, when it comes to the history of science, we don't always pick the right species to work with.
Well, pigs are much closer, right? I mean, that's what
The
pigs.
size of the organ.
And it's sometimes just the biology of the organs.
Right,
that matches so
right,
much
but
better.
sheep
Yeah.
I know
Yeah.
that.
Yeah, but at the same time, what about monkeys or
Well,
apes? Because there's
hold-
always been testing on them because they've always been considered very close.
Hold that question. Maybe we'll
Yeah.
talk about, did you read the Nature paper on cloning the rhesus monkey?
I am probably the least scientific person.
Okay, so here you have some applications. Also diagnostics. We can use CRISPR as a very rapid way to diagnose a variety of infectious diseases like the pregnancy test. Put it on, bing bang, you have it done, it's fantastic. And it's cheap, it's really cheap and fast. And of course, diagnosing cancers, genetic diseases. So they call the diagnostic Sherlock, which I love the name, you know? By the way, does that know what CRISPR stands for?
No.
Not for.
Will you tell us?
I will, don't worry. So here you have some of the work. Now, non-applications of CRISPR, this is very important. What can we use it for? Species intervention. There's a process called, if you want to eliminate bacteria, malaria or Lyme disease, we can use CRISPR in a way called, it's called gene drive. What you do is basically you put in CRISPR into the genomics of the species. So when you want to create a mutation that goes on from parent to child, and it's not like the one gene you're changing, you have the other parental gene. By putting the CRISPR apparatus there, once it's in there, it can convert the other parental gene into the modification. So you can basically eliminate that malaria. How? It's easy. What you do is that, as I'm sure you all know, the malaria is transmitted only by the female mosquito, and the female mosquito are very, very monogamous. Once they mate one, they're not going to mate again. So you put in a gene that makes the males impotent, and you can virtually, they're doing this in an island in the Caribbean, and of course there's problems you can think of, but they think they can really eliminate any species. You can do the reverse. You can revive extinct animals and plants. We know the sequence of the woolly mammoth. We have it down, and they're now using CRISPR to convert that genome to an Asian elephant. Now, they're using to convert the Asian elephant genome to the woolly mammoth. And they're going to create embryos, have the Asian elephants carry them, gestational surrogacy, and we're going to have woolly mammoths. Not for Jurassic Park.
Yeah, that's what I was worried about.
No, because woolly mammoths happen to be very helpful for ecological in Siberia. They have really ecological benefits. And we can revive plants. You can obviously applications for plants, making them cold resistant, pesticide resistant. We can go on and on and on.
Or not.
All right. here's your mosquito, here's your woolly mammoth, and there, we can even go to an extreme state. We know what the genome is of the Neanderthal. So I can take any of your DNA and convert that to a Neanderthal sequence, make gametes out of the woman and the man, and the woman can carry and give birth to a Neanderthal human being. Why would we do this? I'll leave that for your thoughts, but it's absolutely doable. And there are some good reasons that we can do it. All right, so we can use it for designing animals, improve cattle, poultry. We can make synthetic meats. I don't know if you've heard about this. This is unbelievable. I've written about this years ago, but they had a great idea. What they did, they took the stem cell technology, they take a needle, inject it, and remove from a calf neck muscle some muscle cells. They put out a cell sorter. They isolate called microsatellite cells. These are the precursors to all muscles. And they grow these microsatellite cells in culture, and they make fibers. They form these meat fibers. And the first time they did this was about 10 years ago, and the group in England, and they gave it to one of the top 2 culinary experts to taste this hamburger. And of course, what do they say? It's almost as good as McDonald's. And that was at a cost of almost $400,000 for a hamburger. Today, we're down to about $10, and they're doing it all. There's an Israeli company called Supermeat, and they had a really interesting idea. Instead of using it for meat, they do it from chickens, from turkeys, because of course, the largest consumer of turkey is Israel per capita, and they're making these synthetic turkey things. And they really taste pretty good. What's interesting is that you can make it so they're not considered meat. They can be par, they can be neutral, you can make a cheeseburger. We have foods that we can't even imagine that I've never had, which sound terrific. We can change the myoglobin of the meat from, let's say, cow to pig and make pork chops. It'll taste exactly like pork chops.
But it's very energy consumptive, the whole
Yes,
process.
but it's getting better. It is getting better.
Yeah, but the idea was to replace cows, which are,
Well, there's, we'll talk, okay, that's on the ethics side. You're right, all right. And of course, we can make designer plants, as you can imagine, the meat of the future on it.
What about the ethics then of maybe designer children?
Who gets there?
All right, so we've got 3 questions on hold now.
Exactly.
Look at this, these are just some of the companies using CRISPR just in plants, and whether it's cannabis, wheat, cotton, I mean, it's amazing. And we can now make milk proteins in yeast, and that's part of it.
Do we know when we're consuming something that's been modified by CRISPR?
Yes. I think the Department of Health and, Department of, what was, not the agriculture.
USDA.
They know.
USDA.
What's interesting is they do not consider CRISPR as genetically modified. Now that is
That's weird.
That's an
Yeah.
interesting ruling, very interesting ruling. For me, it's kind of a cute story. So on Passover, obviously we don't have any consumption of wheat or even certain types of legumes like corn. We don't eat any corn. And as you all know, Coca-Cola in America, what is the sugar of Coca-Cola? Fructose, which is from corn. But all corn in America is genetically modified. In Europe, they do not allow any genetically modified plants for human consumption. So when they make Coca-Cola in Europe, they don't use fructose, they use
Cane
sugar. So comes Passover, what's Coca-Cola going to do? They have an OUP. So instead of converting all the plants, they import all the Coke from Europe to America, and they automatically have kosher for Passover Coke. So that provides an amazing event in our Seder on Passover. What I do is as follows. I put down Coke, Pepsi, and before Passover, I put down, the Coke from that's not kosher, and cook that's kosher, and I have the kids, how can they differentiate the difference? And they all can.
Yeah.
And from there, I have another one, which is even more interesting. You know, there's different types of matzah. There's hand matzah, there's machine matzah, there's regular matzah. So again, I'll blindfold the kids and give them a little taste of the hand, the machine, or the packaged matzah, and the cardboard box that comes in. And they have to tell us the crazy things. And they always pick the cardboard box. What is that? But this is, all right. So let's review very briefly some of the ethics stuff. All right. There's a time limit. What a time limit means is that you have a right to make a decision, but you have to be knowledgeable. This is not an easy thing. I mean, I'll give you an extreme example. A patient comes in with glioblastoma, which is going to kill the patient if it's 21-year-old within a year. And the doctor says, here's what you have. We can't cure you. These are the various options. What do you want? And he tries to explain to them within an hour what are the options. Do you think that this patient, who's a lawyer or whatever, is going to understand how to make a decision? Not a clue. What does the physician do? Because it's the patient's choice. So is the physician going to give him an exam? If they come back next week and he passes the exam, he can make a decision. No. What is the patient going to say? What would you do in my position? That's not a fair question, because the genetics of the physician and the immunological base of the physician is very different than that of the patient. These are very difficult questions. They seem easy, yeah, give the person autonomy, but it's not always as clear. So that's autonomy. Justice, the idea of distribution. How do you fairly distribute a therapy to the underserved, those who can't afford it? And some of these, as you know, CRISPR to cure sickle cell is $2.2 million. Figured out there are 100,000 patients with sickle cell anemia today. It would bankrupt any health insurance or any company to do that. So figure out what they're going to do. But I'll tell you that in a few seconds. But the idea is that you want to make it available. And you want to make it available not only to your own country, but in a sense globally. As all of you know, if you're reading about, we pay more for our drugs so that other countries can have the same drug being sold at a low price. Is that ethical? I mean, it's nice, but it creates a lot of problems. Beneficence, do good. The idea is, whatever you do, try to do good. It's a very nice ethical guideline, but whatever you do, there's always a mixed bag. It's not always good, and there's non-lessons, try not to do harm. And again, you try not to do harm, but if you are coming in with a glioblastoma, how many people say, oh, I know the side effects, but if I don't do this, I'm going to die, I'd rather take a chance. I mean, these are really concrete ethical challenges what have. And of course, the last one is very much a Jewish one, respecting human dignity. Every life you save is important, and you have to respect this idea of what does it mean to be human, what does it mean to suffer, right? And does a person have a choice? There's a very interesting case. I'm sure you read about this. There was a 17-year-old girl who was had been treated for leukemia for many, many years, and she said she's had enough. That's enough. It's too painful. I don't want to go the next treatment. But she's 17. Parental control. Her parents said, you have to. And the judge ruled, of course, legally, parents had a right, they forced her to undergo treatment. You know, the difference in 17 and 18 is nothing in terms of intellectual, but these are some of the idea of what does it mean respecting human dignity, what is parental rule. I mean, yes, parents have a right to intervene and to say, we're going to treat our embryo one way or the other, we're going to treat our fetus, we're going to treat our child. But again, you know, they have a lot of control over this. And, you know, the child, how many, you know, parents send their kids to yeshiva, or to day school, to public school, or to whatever. They don't ask their 3-year-old, do you want to learn in yeshiva? Do you want to go to day school? It has powerful effects. So it's a very difficult, these are difficult guidelines, but they at least form a foundation by which we can address some of the issues. Here we go. So the 4 Jewish issues I'm going to talk about. Does CRISPR violate any Jewish laws? second one is, are we playing God? Or are we playing, comma, God? Are we enhancing the human condition? Are we using CRISPR to cure disease or to give us blonde hair, blue eyes, which we can do? All right? And the fourth is, under what conditions can we create new breeds of plants and animals? This is a very complicated one. I don't think I'll have to look there. I just want to present it. All right. And
Yes.
designer bodies,
Right.
right? 2020, IQ, etc. All right. So let's look at the rabbinical as well as the first issue. All right. There's a very interesting, first of all, as you can imagine, there is no precedent of this technology in Jewish law, in the Talmud. Nobody's ever thought of changing the human genome. They didn't know about the human genome, so no precedent. But there's a very interesting, there's a rabbi who wrote, called Tferes Yisrael, or Lifshitz. He wrote a commentary on the Mishnah. Very intelligent in the early 1900s. And this is what he says. He says something very interesting. Anything where there is no reason to forbid, according to Torah law, is permissible in Jewish law and needs no justification. For the Torah has not enumerated all permissible things, rather the Torah focuses on forbidden laws. What he is saying is that we can embrace technology as long as the protocol of technologies does not engage in any forbidden action. And CRISPR does not. There's nothing really that we could argue about that, but in basic sense, and there is this moral obligation to save human lives, and that justifies the use of a technology. So when I was in Phoenix, there was a whole discussion, are there technologies that we should not get involved in? Now, in bioethics, there's a principle that I didn't tell you about, that just because we should, that we could, doesn't mean we should. We don't have to. Now, I'll give you an example of what I personally, if you ask me what technology am I fearful about, that's synthetic nucleotides. We can make synthetic letters of the genome. Now, why are we doing this? If you use them for biocomputers, that sounds great, because instead of having an I and O, you have 8 algorithms opposed to make. You can flask of a DNA apparatus. You can store enough information that's encoded by all of the internet in a day. It's amazing. It's slow, obviously, but it's there. So that makes sense. But the idea of incorporating these foreign synthetic nucleotides into an E. coli, which has been done and replicates, scares me because, as I told you earlier, those processes of looking at the errors of the mutations, they won't recognize these errors. And there's no way to ensure that the genome is going to be maintained the way you want it. And that scares me from a bioparasitic perspective. So on that side, there are technologies. We had a big debate about AI. Is AI a technology we should not go into? It was a huge debate. That's another time. So this is basically the idea that if the technology has a good purpose, there's no reason to forbid it because it needs no justification. There's a reason to use CRISPR. As I told you, we can use CRISPR. We have their side effects for curing diseases, for diagnostics, and there's nothing wrong with that. Let's do it.
Professor, ethically speaking, how, oh, no, don't talk.
No?
Save the question.
Ethically speaking, how is CRISPR to cure a disease different than taking a pharmaceutical remedy, like a pill or something like that?
Okay. Because we can not only cure the person, we can cure all future descendants. We can go to the embryo or the gametes and make sure that they never come down with cystic fibrosis.
Got it, thank you.
Okay? That's
Also, they don't have to keep taking the pill.
right.
That's,
Although,
One and done.
yeah,
once it's done, it's done.
generally, hopefully. All right. So the idea is that the other thing one has to recognize is that in Judaism, we have a very important principle. we are partners with God. That God did not finish the creation of the world. There is a gap. And God, basically, from the biblical perspective, says, human beings, I want you to improve on this world, become partners with us. Make sure that the world is safe, is not only safe, but is ecologically safe and sound. And the idea of that we have dominion over the fish and seas and every living thing is to use all our intelligence and our knowledge to make the world a better place, to preserve the environment and fight disease. So we are partners. So the idea, and to me it's a little ironic that the secular bioethics starts saying, oh, we're playing God by using CRISPR. I'm going, what? Why do you bring God into the equation, right? You're a secular, but they do. Anyways, but from a Jewish perspective, it's very important. Now, is CRISPR foreign? So, as again, at this conference, they're all saying this is foreign technology, it's only used in bacteria, and as many of you I'm sure know, that's not true. All of your cells use CRISPR. How? And when you take when you make a message RNA, it's this heterogenic, it has introns and exons, and the cell has to remove all the introns by a spliceosome. It's a brilliant process, and the spliceosome basically has a guide RNA, brings in this enzyme, and cuts, like CRISPR, cuts out the introns. So that, all of our cells have CRISPR technology. Not only that, we make we have about 25,000 genes, but they don't comprise about 2% of the human genome. What's the rest of the human genome? It's not junk. God doesn't create junk. It had a lot of it encodes for message, microRNAs, different types of RNAs that regulate gene function. The microRNAs, the way they work, or silent siRNAs, silent interfering RNAs, is amazing. It creates this small RNA fragment, takes it from the nucleus into the cytoplasm, and engages it with dicer and other proteins, and creates a guide RNA and another protein to cut it, and goes to the message, and then finds the message that it wants to inactivate, or the messages, depending on what, and will bind to that promoter region and cut the message.
I think, just because I'm listening to all this, I think it would be very helpful if you, one more time, just define how RNA comes from DNA. What's that whole relationship? Because I think it's going to help. Because this is, I love this, but it's probably going to go over the head of some people that don't understand exactly.
Good. I'm glad you
Yeah.
said. So basically,
It's my teacher and me. Sorry.
So most of your DNA is in the nucleus. Your mitochondrial DNA, that's a whole different story. We can talk hours about that. But you have the DNA in the nucleus. It's wound up in histones, it's compacted, and when you need a protein from a certain gene, right, there are transcription factors and other proteins come in that allow what's called the RNA polymerase. It's a protein that will convert the DNA to what's called a message RNA. The message RNA has different sequences, I call it introns and exons, and that's very important because we only have 25,000 genes. We have over 200,000 proteins. How does one gene make different proteins? So by selecting which exons and which introns you remove, you have a way of having one gene encode for many different proteins. Once it makes the complete message, it leaves the cytoplasm through nuclear pores, which is amazing. That was discovered by Fred Maxfield at Rockefeller when I was there. Amazing guy.
If I may, it's kind of like a bowl of M&Ms. They're all different colors, and that bowl is the gene. And you can take two reds and a green and a brown, and that is one protein. Then you can take a brown and a red, and there are all these combinations, and of the very large genes, like dystrophin, which muscular dystrophy is involved with. Literally, a gene can make hundreds of slightly different proteins.
And proteins are your molecular machines in your body. These little machines that are actually doing the biology.
Really, I wish I could show you movies. If you saw how these proteins survey and they jump, these transcription factors, they jump over things and they really survey your tumor suppressor factors, the would survey all your DNA to make sure there's no mutations. If it finds the mutations, it stops, and it will stop cell division, cell cycle, to allow it to repair. It's amazing.
There's actually a very simple reason why there are so many kinds of proteins you can make from a single gene. Because the brain, for example, needs different forms of the protein than the kidney or the liver or other organs. And this gives you the ability, this gives nature the ability to tailor specific variants of proteins to different organs.
So I like your analogy of M&Ms.
I love the M&Ms.
Can I steal it?
And
It's
then,
plagiarism.
yeah,
For a
and then
fee.
I'm gonna let you go, but I just wanna say one last thing, because I think this will clarify for everyone here, because I actually love this talk, it's great. Just think about that RNA as a code to make your machine. And in these diseases, there's something little wrong with that code, so your machine has a broken part. Maybe part of that machine doesn't work, maybe the whole machine doesn't work. So by taking this editing and this ability to edit that code, you can fix that part of the machine. You can fix the whole machine, and then you can carry out that biological process that wasn't working, or create, these personal traits, that we talk about.
So let me tell you something about Fred Maxfield, which you probably don't know. Fred Maxfield was trained in Dresden, Germany, came to Rockefeller. He is your typical German professor, straight, very strict at Rockefeller, and he was so strict that when he took on grad students, he would put 2 grad students on the same project, and the first one to get a result, they got the project. He was very, very strict. I had a friend who was an MD PhD at Rockefeller, very bright woman, and she decided to work in his lab. I said, "You're crazy. I mean, do you realize?" And she was an Orthodox Jew, and she said it was fine. She was working on plasmodium, on malaria, and protein synthesis. And when we were there, he discovered what these pores from the nucleus cytoplasm, which are amazing, that are like a doorway. When you have a complete message, they are specific proteins around it that bind to that message and push it through. It's amazing what he discovered. All right, so she's working 5 years, doing some really great stuff, and it's right before Passover, and at that time there was very few computers. Everybody had lab notebooks. You know what a lab notebook is like? And you were not allowed to take them home. You had to leave them in the lab. But she was going to be away for 2 weeks, and she had to write a thesis, so she packed all her 5 lab notebooks in a shopping bag, put it in her trunk, did some shopping for some clothes, and on the way home, one of her best friends lost her father, so she went to pay a shuttle call and she went to the west side, parked the car, went up, paid her respects to her best friend on the loss of her father, and came down to the car. The trunk was open, all the shopping bags were gone. She was a disaster. She came in the next day, said, "What am I going to tell Fred? He's going to kill me." So she comes and tells Fred what happens, and he goes, "Roz, don't worry. I'll hire a technician. You and I will sit down. It'll take a year. We'll repeat the critical experiments. I'll get you out of here. Don't worry. I have your back." Amazing. She really literally cried because she couldn't believe how sensitive it was. And 2 days later, she's walking in the halls, and she sees a little poster on a little note. "Did anybody lose their lab notebooks? Please come to NYU." She ran down to NYU, went to the lab, and there were her notebooks. She said to the professor, "What happened? How did you get them?" So this is what the story became in action. The thief went into the car, took out all the shopping bags, and hailed a cab. Went through the shopping bags, of course, took the food, took the clothes, but these notebooks, you know what they were, he left them in the cab. The cab at midnight came back, was cleaning out his cab, he sees a bag full of these books. Didn't know what they were. They looked sort of medical, scientific, says, "Did I take anybody to any of the medical schools?" And remember, he took somebody to NYU. So he went to NYU, and of course went to the guard's desk, showing him the lab notebooks. The guard didn't know what to say, and just then this professor came by, says, "What's going on here?" They explained to him, and he looked at the lab notebooks. He says, "This person's working on malaria, plasmodium. There's only 4 labs in New York City," and he went to each one in Einstein, one Columbia, one Rockefeller, and one NYU, we put up a little poster, and she got her medicine. Right. He got Fred Maxwell, I mean, this is unbelievable. He got the Nobel Prize for his discovery, which was $1 million Nobel Prize. And she asked him, what are you gonna do with all this money? He says, I'm gonna rebuild a church and a synagogue that were destroyed by the Nazis. Interesting. That's what he's doing. It's remarkable. Anyways, that's a short diversion. Anyways, so we're partners. Okay. So,
What is with the technology?
all right. So this raises a very interesting question. According to Jewish law, are we allowed to endanger our lives by volunteering for a clinical trial? We know many examples, the first clinical trials can be very dangerous. Are we allowed to do this? We can't, I can't harm myself. It's biblical prohibited to cut myself, to do anything that damages myself. Can we do this? And the answer is, of course you can. Because I can choose to be a firefighter, a policeman, because I'm saving other people's lives. And we encourage you, if you have that psychological desire, then that we can enhance, even expose yourself to health risks in order to help the greater good. So that's a very profound Jewish perspective on even now, do patients have the autonomy to refuse CRISPR treatment because of high costs? So this is interesting. I told CRISPR is very expensive, as you've heard. What do we do? So there's a couple of interesting things. CAR T, that's chimeric antigen receptor technology, T cells, is about $600,000. It's a very effective cure for patients with leukemia. How do you pay for that? So you could rationalize saying, well, if you figure out what the cost is to care for these patients under conventional therapy, it's much higher, it's about $1.1 million. So in the end, the healthcare insurance companies are going to make money. But what Novartis, who did it, came up with a very clever idea. They said, you don't have to pay for CAR T if it doesn't work. You have 3 months. If it works, then you pay for it. And if it doesn't work, you don't have to pay for it, which is a very clever idea. And now they're talking about the idea of a mortgage. That just like we pay over 30 years the price of a house of $2 million for your price, $6 million. But anyways, you pay for the cost of the house, so too maybe the government will allow the health insurance companies to pay over the course of 20 years what it takes to cure all these people from sickle cell anemia, et cetera, et cetera, et cetera. So this is in the works. Now, what about the patient? If the patient can't afford it, does he have the right? So in Jewish law, we have what's called limited autonomy. A patient has the right to refuse a treatment if it is, of course, painful, stressful, and it may not prolong life for more than a year. If it doesn't prolong life for more than a year, a patient has the right to say, no, I don't want to do it. If the treatment has a chance, a viable chance, and that's a good question, what does a viable mean? Is it 10%, 20, 30? That's a good question. And then again, that has to be assessed on a case-by-case basis. Then if it's gonna affect your longevity for more than a year, then a patient has less rights to refuse.
Why a year?
There's a principle in Judaic law called Har Shalom and Har Shalom, that a year is considered a very substantial demarcation of longevity, living for a year or 2 years, as opposed to months. Now, the FDA is faced with a huge problem, because here I have a drug that works in 20% of the cases, a lot of side effects, and only prolongs life for 3 months.
Right.
What do you do? I mean,
What
these
all
are not
drugs
easy.
do.
These are not easy questions to address. Yeah, if it's a cancer that can save hundreds of thousands of lives, even 10%, 10,000, that's significant. These are very difficult questions, and it becomes very difficult. But again, just remember that according to Jewish law, there's limited autonomy, but there is autonomy. Yes.
Right, so obviously because of where we are, your background, we're talking about Jewish bioethics. But do you ever have conversations, I mean, you talked briefly about when we're talking about the pigs, and that people who are Muslim or follow Islam have the issue with, why you're doing this with pigs. But are there other religions, Buddhism, Catholicism, different sects of Catholicism, that you also have these bioethics conversations with? And, just out of curiosity, like with the discussion like this about the autonomy refused, do you have those, where do some of the other religions stand on some of
Okay,
this?
that's right. So we have this biosec program. We really expose our students to Buddhist, Catholic, we have strong Muslim contingency. And it's interesting. I was invited to be a witness for the National Academy of Science, the highest science academy, on what's called human-animal chimeras, neurochimeras. What is the ethics of taking human neurons and implanting them into mice or monkeys and giving them human intelligence? So it was a very interesting story. There was myself, there was an Islamic woman, a scholar, and there was a Catholic scholar. And it was on Zoom because it was during.
Do they walk into a bar?
Anyway,
Especially on St. Patrick's Day.
so what's interesting, the Catholic priest did not show up. So it was me and the Muslim woman. And it was very interesting that there's a lot of similarities. We are, in a sense, based on Abrahamic philosophy. So there are differences and there are combinations, but we can have these conversations. We have some very good Islamic scholars, physicians at New York Med, and we do cross-cultural bioethics. So we have courses where we discuss that. Understand, just like in Judaism, you have Reform, Conservative, Orthodox. In Islam, you have Shiite, Sunni, you have Southeast Asian. There are different views. I have to tell you my impression. There isn't the same, at least my experiences. In Judaism, we have a very concrete process to do this. It's less concrete in Islamic. In Catholicism, it's very concrete. The Vatican is very interesting. The Vatican, I don't know if you know this, every few years has select conferences that invite experts from all over the world to describe to the archbishops and to the cardinals what the newest technology is. And they use that. But the Catholics have their own rigid rules. You cannot generate a human being without marital relationships. And look at Alabama, what the result is. So there are different views. It's interesting, the Buddhist, do you know about karma in Buddhism?
Yes.
Mm-hmm.
So what that means is that if you do something and there is positive effects and negative effects, one doesn't take over the other. You're responsible for both. So if you do something that has a positive effect, you save a life, but on the other hand, there's a negative effect, you're guilty for both of these. So it's a very interesting type of philosophy. Yes?
My question's really, if it's not life and death that CRISPR's doing, but just quality of life. And I'm not talking about blonde hair, but just like.
Right, why not? Why
Is
not?
it the same thing with
Yes,
the year
yes,
and
yes.
okay.
Yes, yes, it is. Quality of life is very important in Judaism. It is. All right, so, so we've talked a little bit about time and justice, and again, in Judaism, we want to share our knowledge. What's interesting in Thailand, which is striking, when they guarantee all the surgeries in these hospitals. And so they do a lot of breast augmentation reduction. And if you know anything about breast surgery, in America, probably about 6% of the time they have to go back to do surgery. So in Thailand, with EMP, they said we've got to reduce that. It's very expensive. They spent 4 years developing new technologies of breast augmentation reduction. It's amazing. Why don't you publish it? Publish it? No. It's our intellectual property. They won't publish it. They have, now this goes against Judaism. Judaism says, do you have something? Get it out there. Make it available. So it's very different cultural things. The funniest thing is they have, he has 20 hyperbaric chambers. because they have evidence that if you take autistic children and put them in with their mothers from 6 months to a year old, for an hour a day in this hyperbaric chamber, they see dramatic results. And I've been there for many, many years, so they know me very well. And they showed me the results. And I said, wow, this is really interesting. He says, why don't you publish it? We have experts. Well, help me publish it. We can't do that. It's our intellectual property. Not only that, they're doing it for Alzheimer's disease. They take patients who are early stages of Alzheimer's, they put them in these, and it's a $10,000 machine to put them in. And now, this is years ago, they're starting these clinical trials in America as well. I had a friend who just finished one of these trials. An hour a week, hyperbaric chamber, we'll see what happens in terms of Alzheimer's.
What is a hyperbaric?
Hyperbaric chamber gives 1.3 atmospheres of. It's approved in the United States for, like if you're swimming and you have, you've been too deep, bends the
Okay,
disease,
I got it.
right? Or if you have stroke, you want to improve circulation of the brain.
Also, viral infections like rabies, it increases the amount of oxygen
Yeah.
that goes to each cell.
So it's again, they would not release this information, though I saw it, but I can't do anything about it. It's very interesting about hyperbaric and longevity. We have twins, Scott brothers, who are both astronauts. And several years ago, one went up to the shuttle, right? You know the story.
Yeah.
Yeah.
And their telomeres, you know what telomeres are? Those are the ends of your chromosome. They're non-coding ends, and you're all born with about 15,000 of these base pairs, these letters. And as you age, they get shorter and shorter and shorter. So they looked at the telomeres, and the guy who's in space, his telomeres actually grew. Normally, they don't grow, they get shorter and shorter and shorter. So an Israeli company, of course, was very smart, said, wait a second, why is it in space? Because they have hyperbaric oxygen to get more. So they have started a whole study to put these people into hyperbaric chambers to see if that would improve longevity. So it's interesting, right? Fascinating, right? You know the sound for my course? All right. So, how are we doing, Tom? Okay? It's fine. All right. Okay. So now we go into the second response. The idea of playing God. We discussed that and want to improve the world. What about when we really change the dynamics of human reproduction? The example is making a 3-parent embryo. Okay? As you may know, there are 2 major sources of DNA in the cell: the nuclear source and the mitochondria. You have about 1,000 mitochondria in each cell, and each mitochondria has at least 2 to 10 copies of DNA. There's a lot of DNA. They're small, only about 37,000 letters, and they code for about 13 proteins, but they're critical. The mitochondria is your battery house of the cell. It's very important not only for providing energy, but there are diseases. If you have defects in the mitochondria, cause a whole variety of diseases. And to make something less complicated, just understand that mitochondria doesn't have the same repair mechanisms of the DNA as the nucleus does, but nonetheless, mutations can arise. And by randomness, you could have women who have mitochondria that are defective, and for a random choice, they have more mutated mitochondria in their gametes than they, and of course, mitochondria all comes from the women. So now these women who have defective mitochondria in their gametes, which is, will have children who have severe disease like muscular dystrophy. So they don't want to have kids. So they came up in England with this wonderful method called mitochondrial replacement therapy. And what they do basically is, I'll just describe one method. They'll take a donor woman who has healthy mitochondria. They remove the nucleus, and they transplant the nucleus of a woman who has defective mitochondria, just her nucleus into this donor egg, and then they fertilize it with her husband, and she has 3 parents: the mitochondrial donor, the nuclear donor, and the sperm. So, what is Judaism deal? How does that work? So there's a very interesting, this is a Rabbi very interesting idea. There's a very famous Talmudic statement that says there are 3 partners in the creation of a man, woman, and God. What happens if there are more? Here you have a fourth. So he says that this statement is not talking about a halachic, a legal statement. It's a preferred. The preferred method is this method. But if there are other methods that will be helpful, and what's the proof? Proof is Adam and Eve. Adam was created with no sperm, no eggs, never was gestated. Eve was created from, obviously, stem cell from Adam, right? And with God, only had two. And what does God say in the Bible? By Har Shalom, he named them human beings. So he's saying a very important lesson. That according to Judaism, as long as you use a human cell, a human DNA to create a human being, that by definition is human being. And even though you have 3 or 4, parents, we can have 5 or 6. As long, and what's really interesting about that is that we know that I can take your stem cells and I can convert them into gametes. So I can take a man's blood cell, a woman's blood cell, convert it to gametes, make a pre-embryo, plant in gestation. There's no sperm and egg. Is that a human being? But according to Yeshua, it is a human being. It was started with human cells, it's a human being. So that's a very important insight.
What's the legal status of this currently?
What do you mean?
Is this permitted in all states?
This, not yet.
Not now that one.
Yeah, that's what I thought.
It's only, you get it.
Yeah.
But Columbia has frozen many of these. We're hoping it'll be open. And it works.
Yeah, no, I know that. I'm just,
Yeah, but it's,
I know a number of people who've actually, there's a black market in this in the United States.
really?
Yeah,
How much they charge you?
I don't know. No, I don't know.
You come with only half the information.
Bitcoin.
But there is definitely a black market that gets done in this state.
Wow.
Well, it's so interesting because, stem cells, except for certain conditions like bone marrow transplantation, is really not allowed in the United States, except in a few conditions. And yet there are 700 clinics doing stem cell therapy. So what do? You need one stem cell therapy to theoretically cure cancer, cure dementia. They send you to Mexico, they do the stem cell procedure, and then they bring you up and they follow you. That's another black market idea. Anyways, so this is the idea. Okay. So let's talk about a third ethical issue. Come on. All right.
That's my question.
So, right, enhancing the human condition for creating designer babies. Let me tell you something which I find interesting. Since IVF was processed from first test to baby in 1978, Louis Brown, brilliant, Dr. Edwards, that was a brilliant idea. It's really, he would never have gotten approved today, because to do it by FDA, you have to do a 9-year study to look at the effects of this, but got approved. And we've had 12 million babies born. There's no published report of a designer baby. Does this mean that there's a moral compass in the world? That we're not going to use this technology to create designer babies? We'll use it to create a child that has to give a bone marrow transplant for an older child who needs one. Maybe, but that's not really the idea of creating a child with a perfect IQ. Yeah, we have 100 genes, right? That
You really don't know how to do it.
What? Well, we have 100 genes that we know affect IQ, and we can modify them with CRISPR, presumably, to enhance IQ. But again, what is the response to this? So, again, does it violate the prohibition of harming oneself? All right. And here we have a real interesting model, plastic surgery. Plastic surgery is enhancement, right? Dyeing one's hair, removing wrinkles, doing everything. And the Jewish response is that, you know what? If there's a real psychological need, go for it. There's no problem. You're harming oneself. Yeah, you shouldn't do it. I should not do facelift or improve my face so I can mingle with younger women. No, that's not the right idea. But if I need to, because I feel I'm embarrassed, because I'm aging too quickly, or I look too old. I'll tell you an interesting story. There's a very famous rabbi, Farkstein, he was from Israel, and he used to come visit in America. Wonderful man. And one time I used to meet with him like an hour. Brilliant guy. So I was giving a lecture on facelifts and, you know, plastic surgery. And I said, what is a Jewish response? He says, look at me. What do you see? I said, why? He was about 50 years old. I said, one of your eyebrows is completely white. He says, yes. I was 20 years old, and I woke up one Saturday morning, my eyebrow turned white. I was embarrassed because the Gemara and the Jewish law says you cannot dye, a man cannot dye his hair from gray to white. It's a law, right? He said, what am I going to do? So he went to the head rabbi, who was called Chazanish, very famous rabbi. He says, what am I supposed to do? He goes, what do you mean? Dye it. After Shabbos, put some dye. You can't walk around like this. It's embarrassing, you know? If something is embarrassing, if you're a Jew, and again, the rabbis will tell you on a case-by-case basis, if you're doing this because you look younger need to look younger for your job or for whatever, that's legitimate. So again, if CRISPR is being used for a legitimate psychological reason, there's not going to be a problem, but it has to be assessed on a case-by-case.
So with the baby, so you're not going to specifically choose an embryo that's blonde hair, blue-eyed, you know, high IQ, but you can maybe pull one out that would have cystic fibrosis and not implant that one.
Right.
Or
The idea for legitimate psychological reason implies consent.
Exactly.
And an embryo can't consent.
But the parents.
Yeah, but that's an interesting question, because embryos grow into children, and children have sued parents
Of course.
Right.
for things.
Right.
So, I was born in Sweden. You like my blonde hair, blue eyes, 6 foot nature? So the question is, can a parent says, I'm moving to Sweden, can I use CRISPR? I have Ashkenazi background, can I use CRISPR to fit into society? It's a very interesting question. It's not easy, it's not a flat no, it's not a flat yes. One has to really look at it on a case-by-case basis. And that's, I think, what?
Well, I wanted to tie into also what Jody was saying about the designer babies, that then it goes to what Jay was saying about kids suing parents. These parents who have one child who's born who's horribly sick, wanting to specifically choose an embryo where that
Right.
child is then going to be able to done.
At
There are a
risk,
lot of them. There are a lot of them.
and I don't want to take your time up, but it's getting very, very complicated. So for example, autism, we now talk about a spectrum. And a lot of people who perhaps are on the spectrum consider themselves non-neurotypical. And they think this is an okay way to be. And that anybody who alters them is doing them harm psychologically. So we know a person with Tourette disorder. He's probably the world's greatest soccer goalie. He's certainly the best American, Tim Howard. He was our World Cup. And he said his Tourette's gives him special abilities, quickness.
Okay.
And he said he wouldn't change it.
Look at Einstein.
I'm sorry?
Look at Einstein, Beethoven.
Yeah.
They all
Yeah.
probably had.
Right, but
So,
the deaf community too doesn't
the
want.
question of what constitutes when you get
Right.
to things that don't result in death, for example. Sick people with sickle cell die early. They don't live, and it's a painful life. So that's an easy decision. Duchenne's muscular dystrophy, cystic
What about
fibrosis.
Huntington's, right?
Huntington's,
He
yeah.
lived for 50 years. I'll give you a case that was in Genome.
Most of the Huntington's patients that are diagnosed don't want to know their
Wait,
diagnosis.
listen to this case.
That's a very
Right, a lot
interesting
of-
story
So,
there.
I'll give
But-
you an unbelievable case that was in JAMA, Journal of American Medical Association. There was a man who had a family history of Huntington's. His father died of Huntington's disease. It is a dominant trait. So if you have that gene, one of those genes, you're gonna die. And depending on what's called repeats, you can always predict to the month when you get the first symptoms. It's very predictable, but for the first 50 years, you virtually have nothing detectable in terms of symptoms. So he said, I don't wanna know, because what Jay said, there's a high risk of suicide. People know this. Who wants to know you're gonna die when you're 50? It's a terrible disease. So he said, now don't I wanna know, I don't wanna get married. But he graduated, and he fell in love with this girl. And the girl says, let's get married. He says, but if we get married, we can't have children, because I don't want to test the children. If I test the children, I will know whether I am, I have the disease or not. She said, don't worry, we'll adopt, won't have children. As fate would have it, she accidentally got pregnant. What does she do?
Test the baby.
Does she tell him? Because if he tests the baby is positive, they may terminate the baby. Because he would say, terminate. And then he'll know that he's dead. You know what she did, right? Listen, she lied. She said, I tested the baby.
And he's fine.
Was fine. Why? Because she said, the baby's not gonna come down for 50 years. By that time, he's dead. He's never gonna know. If he has it, he'll know in his 20 years. And we have 50 years, who knows what new discovery is going to happen. Isn't that an interesting solution? Very clever. Anyways, so, again, oops, sorry.
But what about designer babies they're doing in China that are resistant to HIV, but they
Is that
don't?
really designer? No. Dr. Hay is doing this experiment, did it because they came from parents who were very, very struggling with HIV and wanted to make sure that these kids would not have the same struggle. It's not classical designer babies. I don't have any report. But what's interesting about that case, I love that case. Why? Not because what he did was ethically wrong. He didn't follow protocol. It was illegal. He didn't follow most updated use of CRISPR for doing that sort of stuff. But what it did, it brought the world's attention to all the ethics of CRISPR. And that, to me, was very important. So he never went to jail, by the way. He was convicted, but never went to jail. All right. So Judaism values psychological status of the individual. That's the important lesson here. So, the third response. All right. So Maimonides says the following, that wounding only applies when it's done in a contentious manner. And cosmetic surgery, as I told you, done to undo something that is suffering for bad appearances is completely permitted. That's a very important thing. And even pierced ears or liposuction, when there are legitimate psychological concerns, go for it. All right? So, now, what about enhancing longevity? We know in the Bible there is this idea that people should only live for 120 years. There's a time to be born, a time to die. What do we do with that? This is really not discussed by the rabbis yet, so I'm giving you my impressions. As I said, I'm not a rabbi. But basically it works as follows. There is in Jewish philosophy the idea that prolonging life as an opportunity to better serve God or mankind and make it a better place is valuable. So if there are limited side effects and you want to do this, then there is room, at least in my opinion, to say, let's go for it, right? It's very interesting that in Judaism there's very interesting philosophy, not universally accepted, like, can you retire? Can you retire in Judaism? And there are those who says, no, you know it. You're here for a purpose. You have a purpose in life. You have to discover that purpose. We say that in our prayers, that we thank God for making us aware of what our purpose is. So I had a very interesting case. I have a colleague who's a world's expert in low vision ophthalmology, really brilliant woman. She deals with a lot of these cases, and she's also a great scholar on the Bible and the Talmud, and she gives a lot of classes on that. And she goes to her rabbi, she says, I want to retire from ophthalmology, and I want to start giving classes and start being teachers and rabbinics. And the rabbi says, you can always give classes, but you can't retire. Because your purpose in life is clearly, as this expert in ophthalmology, we can't lose you. So there's a very interesting idea of what, philosophically, you have to know what you're here for, right? And what you're here for, and to engage in that. It's very interesting that when I was in college, I had a rabbi Aaron, one of the most brilliant rabbis I've ever met. Really, he had a photographic memory. He took what's called the pin test. You ever read about the pin test? The Talmud is a fixed text, and every word is on a fixed page. And the pin test, you put a pin in one word on page 1, and what word does it go through page 2, 3, or 4?
Oh, geez.
At 16, he could do that. He really had a photographic memory. He was a great professor of Tom. And my senior year, he calls me in and he says, Blakey, what are you gonna do with yourself? I said, I'm gonna go to science. He said, no. You should be a rabbi. You should be a teacher for rabbinics. I go, I'm thinking, oh my God, in this class of 70, I was average, really, maybe. Who is he talking to? He's mixing me up with some of these brilliant people who are sitting in the front row. You know, in the Talmud class, the closer I to the Rebbe, the more brilliant I was in the middle. So, and he spent 2 hours trying to convince me to go into rabbinics. And I said, Rabbi, I don't have a brain, I can't do this. This is not my thing. He said, no, you'd be excellent. 20 years passed, and we're having a reunion, and with all the other 70 students, and I said, at that point I figured out, he probably said that to all the students. He wanted all the students. So I'm sitting around and I said, all right, how many, look, we only had, with 70 students, only 3 went into rabbinics. Everybody else went to doctors, lawyers, financial people. They said, what are you talking about? He never called me to this office. So I was very confused. And one of his students said, John, you don't understand. He was not only brilliant in terms of academics, he understood his students very well. He obviously saw in you a certain talent of teaching and wanted you to pursue that. Which is maybe true or not. But anyway. So at that point, my daughter said, you better start teaching. That's how I took the job at Columbia to teach. Because, all right.
Oh.
So, what about modifying and creating new species variants? So, we can do it. The question is, this is a very, again, I don't have a rabbinical answer from an expert, but I'll give you my impression. Species are extinct for 2 reasons. Natural extinction, because of normal evolution, and because of human intervention. We kill too many cheetahs, we kill too many rhinoceros, right? So if we kill them, maybe we have an obligation to revive a species. And I will be in favor of that. If a species dies naturally because of evolution, I'm a little more hesitant because that's part of how the world was created. That species come into existence, they survive and some die. So I would argue with you that there may be a difference in destroying extinct species due to human destruction versus natural selection.
So no Jurassic Park.
What?
So no Jurassic Park. Good.
Yeah, so then how do the woolly mammoths fall in that? Because if they died out because of natural extinction, and I know you said that they would bring a lot to like Siberia if they were
I
found.
don't know.
Well, they were probably killed by humans.
Oh, yeah, the mammoths probably were.
Mammoths were probably
Yeah.
killed by
Were they?
humans.
Right, not the
Yeah, they're
Jurassic
hunted out
Park,
by-
yeah.
Okay.
But
Again, these are very good questions, but it requires a lot of thought, a lot of discussion.
I think every year, several thousand species go extinct.
Yeah.
And we don't even know about most of them.
Right.
And to focus our energies on, and our money, on bringing back one species, considering all the other things we could do that would bring good into the world, I have very serious ethical concerns.
Now, he's raising a very important point, which is how do you allocate monies?
Yeah.
This is a very, very important point, and beyond the scope of any discussion here, because it requires not just rabbinical intervention, it requires experts in finance. I mean, this is an excellent point, and maybe you'll let it pick her
Well, maybe we'll stay here for the next 3 hours after.
All right, what are the
humans don't go extinct.
Well, by the way, you know, it's very interesting, you know that in many yeshivot, they don't want you to teach evolution. You know this. You know, but it's really, it's overplayed. Because when I go into a yeshiva to teach about evolution, I say, if it wasn't for evolution, we'd all be dead. Literally. We have specific immune cells, T cells and B cells. We have about 1 million of them, and each of them recognize a certain protein, what's called epitope. So if you're exposed to this pathogen that has this foreign protein, it says, I'm foreign, then the immune system kills it. Sometimes we're exposed to a pathogen that we don't recognize as foreign. So what does our body do? Our body, these cells that make the antibodies, will self-mutate in a certain region of the antibody production, the variable region until they get the right antibody to recognize that pathogen and cure us. So this evolutionary process of self-mutations, part and parcel of all of our lives. Just throw that in all over. All right, so correcting genetic mutations opens the door to a whole area of genetic counseling. Because now, no longer does a couple have to say, terminate, don't terminate, maybe undergo CRISPR. It's a whole new area. Also, the idea of human-animal neurochimeras I talked to you briefly about, that can we create monkeys, mice that have, when I presented that to my students at Columbia, they said, well, that's a good question. Does Columbia admissions have to accept a monkey that has 40% of its neurons human? It's a good question. That's all I was saying. But anyway, these are really important things. And as I spoke to you briefly about xenotransplantation, I'll tell you my opinion about xenotransplantation. I don't think there's any problem taking a pig heart. I mean, we do this all the time with other organs, valves, heart valves we take from pigs, and other things in the eye. But I think that the future is not xenotransplantation. I think the future is organoids. And that is taking the stem cell and creating an organ out of the stem cell. I think that's where the future will be. And we avoid many of the ethical dilemmas of xenotransplantation, etc.
For the first one, is that kind of allowing the embryo to heal itself through CRISPR? Is that,
Well, we intervene.
right, intervene, and then the embryo with CRISPR kind of heals whatever's going on, so it can be a healthy baby.
And I think, all right, so this is my conclusions. All right, that there's a great deal of potential in CRISPR technology. It's amazing. It's so new that there are not many rabbinical articles that discuss the legal or ethical ramifications of this technology. We're working on that. And the problem is, I don't really know modern day rabbis, at least in the Orthodox world, who have the science background to study this. I'm going to be in Israel over Passover. There's a rabbi there who's he's very brave, and he's very well accepted. I'll meet with him. I'll see. He's very well educated. I'll see if he'll be willing to take my course in genetics and address these things. All right. We need, part of this is that we need to educate, both in our schools and our yeshivas, this whole purpose, that this whole science is really undergoing revolution. With stem cell technology, CRISPR, all this sort of stuff, we have to be better prepared. And just like we did this with medicine, we did this when Jacobowitz from England started medical ethics in England, and Tedler in America started a whole course on medical ethics to allow physicians to really understand both the Jewish perspective and the medical perspective. We have to do the same thing in our education system to develop quality people who could address these issues. And that's basically what I have to say.
Yeah, that's great.
That was really excellent. I have a million questions, but just a very general question. One of the problems with Judaism is that there's no Jewish pope. And as you well know, you're standing in a Reform congregation, and I know the difference. I was raised Orthodox. I doubt there's really one opinion on all subjects.
I think that's true across the board. I agree with you.
And it's hard to know.
It's good. I think it's good that there are different opinions. And I think it's up to the public and other scholars to understand how these opinions are formed. I mean, how are you going to argue with the church, right, that you need marital relations to create a human being, and that sperm and egg in vitro is not a human being, and yet on the same side, destroying that is murder. So I think this is what culture is all about.
Yeah, and I think personally Jews have it right. Because if someone doesn't like the Pope or what the Pope stands for, they leave Catholicism. The Jews just changed synagogues.
It's like the story of the, the man caught on a desert island.
I just heard this one.
Right? And he had two synagogues.
Yep,
One that
I belong to two. And there is one I wouldn't step in.
But no, that's a fair question. So any questions about CRISPR, anything in technology that you want to address, I'll be more than happy to.
So some of this is just personal questions. Talk about CRISPR being used for repairing genes, for diseases and things like that. Is there anybody who studies CRISPR in terms of repairing nerves?
Nerves?
Yes.
Of course.
And is it being used technology-wise, or
Okay. I don't know maybe you know what the latest is on.
I'm not sure I understood the question.
No, it's used to
Well,
prevent neurological disease.
Oh, yeah.
For repair.
The problem with any genetic disease in an adult, when you have an adult patient, is delivery of the whole CRISPR system to the cells that you want to deliver it to. In this case, a neurological disease usually involves the brain. And the brain is enclosed in a peculiar box.
Yeah, but it's not
That doesn't give
neurological
us much
genetic.
access.
It's talking, where, using me as an example, I've got the situation I've got right now because my sciatic nerve it was impinged by my nerve.
That's different.
And that's where I'm looking, is
That's,
the DNA.
and I can actually help you with that. I'll talk to you after.
That's true. Almost, I'd say 95% of all human conditions, except for trauma, have a genetic component. Right?
Okay.
But trauma, when you break a bone, even that has a genetic component to it. HIV has a genetic component. There's a variant of a protein that if you have the variant, the HIV virus does not enter your cell. And you're basically immune from getting HIV.
Right.
But most, except for trauma, most diseases that you encounter in life have some sort of a genetic component. So, yeah.
Speaking of genetic components, can it help with osteoporosis? Because that's a bone thing.
I mean, again, bone things are a little bit easier because they're to some extent controlled by bone marrow, which is easily exchangeable. But the question with osteoporosis comes up, are we going to use $1 million, $2 million therapy? Or are we going to give you a pill that we currently have that's going to ameliorate it? The answer now is you're going to get the pill. Maybe 50 years from now, there'll be some kind of instant gene therapy. But, as I said very early, it's a very difficult process. It's a very expensive process. So, only those diseases that have no cures and no treatments are going to be the ones that go first, and there are a lot of them. There are actually 5,000 over 5,000 in the McCusick book. Occasionally we consult on Tay-Sachs and Sandhoff's disease, which are diseases that are more prevalent, not unique to Jews, but more prevalent. And they result in death, certainly by early teens, if not sooner. And that's a disease that the Jewish community, I've been approached by many Orthodox rabbis. That's a disease that they're very interested in gene therapy. But osteoporosis, especially because it's more common among older people, is not likely to be high on the list.
Thank you.
So my recommendation for any disease, speaking as an immunologist, is that I think the immune system is a critical component of health. And whatever you do to maintain a healthy immune system will only help you.
That's right.
What does that mean? It means, ready for this, exercise. And if you don't, I'll tell you what I do. If you don't take if you're not out in the sun, I take vitamin D3 every day.
Me too. 4,000
units. It's really been unbelievable. I do vaccines. I think vaccines are amazing. And they offer things that we don't, there are now reports saying if you take the COVID vaccine, flu vaccine, it lowers cardiovascular events. Did you read about that? All these things, why? Because it's boosting your immune system. And a healthy immune system needs a healthier person.
Well, and that's another discussion, Jews in the anti-vax movement.
So
I know, it really pisses me off.
I have kind of another completely unrelated question.
I'm gonna just,
oh, yeah.
I'm gonna limit
I-
three more,
I-
three,
in all
three,
fairness,
three,
you
Jay,
have to
Jay. Stop.
three more questions. Max, Jennifer, Jesse,
Yeah, he
and then
just-
unfortunately, we're gonna have to wrap this up.
I just have a completely different question from, what's that?
Max.
Max, yeah. In Orthodoxy today, how often are you seeing students who are close to the Torah, who read it a lot, who study a lot, going into science, thinking about these questions? Because I noticed in my classes today, I don't see as many anymore as I used to. It's changing.
I don't see it.
Yeah, and you interest me in that regard because you know a lot about this, and I'm actually quite I'm happy and I'm optimistic to see that some of the liberalness and the thoughtfulness that goes into the rabbis when they make these decisions, but I'm afraid that's going to change if we don't have more.
I couldn't agree with you more.
25 years ago, half my medical class was Jewish.
Where was that?
At Rutgers.
At Rutgers.
And also at Indiana before that. And now, last time I taught was about 3 years ago. There was one Jew in the class, and they were mostly South Asians, Indians, and assorted other South Asians.
And then,
I said to him, Goldstein, you're an endangered species.
by the way, the Asians, the Chinese are
My classes.
incredible students. They're really,
Oh, they're smart.
they're
Right.
smart and they
But it,
motivated. They're like we were, in the 50s.
so I have this conversation, of course, with all of my students at Rutgers, right? And many of them, I don't know if you know, they'll tell you when they came to America. My friend Jigger will tell you we were taught to emulate the Jews. That's what the Indians were taught in India.
That's very interesting.
Yeah, and that's what they would tell you.
Yeah, I've
So
heard
you
those
hear,
exact words.
that's
Yeah.
that's why we do what we do, because we were taught that's how we do it.
Those people should be more
And it's,
vocal.
it's
Like the Jews, I love that.
you know that we have the largest percent of Nobel laureates
Yeah.
in the sciences.
Yeah.
And there's a reason. It's not just that we're intelligent, there's the motivation. I'll tell you.
And City
Yeah.
College.
I'll tell you an unbelievable story. In the Bible, the last 2 weeks, we were learning about the building of the Mishkan, the temple, and discusses two individuals who were assigned the task of looking at the architectural plans and building it, and it's caused them, in the terms that are called chachmas lev, wise people of the heart. It's very strange. I mean, to be an architect, you have to have intelligence, creativity. Why is it? It's not just one time, over and over again. Chachmas lev, chachmas lev. So I never understood this, but as I reflected back, when I was 14 years old, there was a very famous rabbi, Rabbi Shlomo Kalbach, who composed a lot of music. And he was very inspirational. And one time we invited him to an NCSY, that was a youth group in Syracuse. He came, 200 kids were mesmerized by his music and his storytelling. It was amazing. As he walked home to his house, they were staying in Syracuse on a cold winter night. One of my friends turned to him and says, "How do you do this? How do you compose all these melodies? You sit down with your guitar and just compose?" He said, "I can't do that. If I'm not inspired, nothing comes to mind. If I'm inspired, I can compose anything." So of course, my friend says, "Are you inspired walking with us?" Of course, then compose a piece. And he did. He sat down 5 minutes and composed a piece. But the lesson is very profound. To be creative, you need that inspiration. If you don't have that inspiration, that motivation from the heart, you'll never be creative. And that's what we have to infuse into our students somehow. All right, any other question?
Jennifer
I
and then
You are
Jesse.
Jennifer,
Jennifer, hi.
you never told us the definition of CRISPR, which I know so much. I wanted to bring it up, so I want to make sure you brought it up.
We can Google
Let's
it.
see. Clustered,
I don't know it.
I knew it, I've seen it.
Chris.
Sorry, did not mean to put you on the spot.
Regulated
Intermediate
interstate
disperses.
short palindromic repeats.
Yeah, I actually don't.
I
I
actually don't think it's important then.
Yeah, I don't
But
know.
you know
it.
what? I love the acronyms. It's like Sherlock.
Yeah.
Do you know what Pac-Man is?
Yeah.
I can tell you, Pac-Man. Pac-Man's great. I'll leave it with that.
Is that an acronym?
Yeah, Pac-Man.
I did not know that.
Use, Pac-Man is a CRISPR. You have different types of cutting mechanisms. Cas9 cuts DNA. Cas13 cuts RNA. So now you have a Pac-Man system. What do? I take CRISPR, a guide RNA that recognizes the cancer mutation of the cancer, and infects it. A nanoparticle infects all of the cells, hopefully, all of the cancer cells. And if it recognizes a cancer genome, instead of cutting the DNA, it cuts all the message around all the RNA. And without the message, the cell dies. So it's a Pac-Man way to kill off all the cancer cells. By the way, your question is a very interesting mouse study with stem cells. If you repair, there's a genetic disease that causes death of these mice within months because it shuts off the hepatocytes in the liver. So these mice die within 3 months instead of every 3 years. And if you fix it with gene therapy or CRISPR, right, and they went back and says, what percent of those hepatocytes were actually fixed? Turns out it was only 10%. This particular disease, 10% of fixing the hepatocyte would cure the mice. Wow. Them to live for 2 years. So I don't, it's going to be varied by disease, but it's not, it doesn't necessarily have to be 100%.
Okay, final question, Jesse.
No, we're trying, it's okay.
All right. This has been wonderful. I hope
Okay.
you enjoyed it.
Yes, thank you. Thank you, thank you.